Thank you so much for inviting me here in this beautiful place. So indeed, our work is focusing on cellular perspective of intelligence. And so I think my talk is a bit different from the previous talks, and maybe also your background is a bit different. So if you don't understand something or want to clarify it, please shoot your questions while I'm talking. It would be very nice to have more interaction. And just to give you a bit of a kind of a context where I come from. So our lab is mostly interested in the questions of evolution and human cognition from the cellular perspective. So we see neurons as computational units of our brain. And of course, in the course of evolution, our brain has expanded quite a lot. And this gray matter expansion, or brain expansion, does come at a cost of energy consumption, connectivity, speed, and efficiency. So it's almost inevitable that instead of just inserting more and more neurons or computational units in the brain, we need adaptation. Our neurons also underwent adaptations. So to make these neurons kind of more efficient, to let them perform maybe simple operations in a faster and more efficient way. But also we think that human neurons can sometimes play a role of a small network where one neuron can perform computational operations that can be otherwise performed by a whole network of simple neurons. So in our lab, we're actually interested in two big questions. And one question where we started with is, do human neurons have specialized properties? Are they different from other species? And while researching this question, we came to a realization that indeed there are some features that are human specialized. And we would think that maybe those features also underlie something that relates to cognition of humans. Maybe they make human species also like cognitively more capable. But to answer this question is really difficult. And then we came up with a different question. So once we find those human specialized properties, we asked, do these properties also matter for human cognitive ability if you look within human population? So do these specialized properties, can these properties explain differences in intelligence between individuals? So that's kind of how we rolled up in this research. And of course, I'm very happy and grateful to Kirsten and Adam that they already explained quite a lot about intelligence. And indeed, intelligence is something that we can, is very diverse in human population and can be plotted in this bell-shaped curve where most people will have this average intelligence, some will be outliers, have high or low intelligence. And we also know from big statistical research that intelligence does correlate with a lot of life outcomes, such as education, career, socioeconomic status, but even health and life expectancy. So intelligence does say something that lies at the core of the differences between between individuals in their personality. But what about the neurobiology behind this trait? And again, intelligence is a very, very complex concept. It can be defined in very different ways. And we took a very reductionist approach to this matter. So we define intelligence as something that can be measured by IQ tests. So, of course, outside this test, there is a lot to say about social, emotional intelligence, but we cannot measure it in our research. So we will just disregard it and stay with this reductionist approach. But despite some controversy about IQ tests and how much they can tell us about our intelligence, we do know in the approximately 200 years of IQ studies that they do say something about general level of intelligence. So many of cognitive tests, as also Adam was explaining today, many different cognitive domains correlate with each other, and together they create this general factor of intelligence. these test results, the cognitive test results, are also very stable in time. So there are also studies that show high correlation between tests that are taken at the age of 11 and then the age of 80 years old. So although these are not the same tests, they do show high correlation, so they're stable in time. And finally, there is also very high heritability of intelligence, up to 80% in the adult population. So the IQ tests, despite all this controversy, do say something meaningful about our biology. And there must be something in the brain, in our brain, that can explain the differences between people with high or lower IQs. And again, if we look at different approaches to intelligence, how can we study intelligence, we can actually see that most studies kind of use these two opposite strategies. And one that we heard a lot about today is to study the brain, networks in the brain, areas of the brain, their structure and function, and how this relates to intelligence. And that also provided quite a lot of insights and areas that relate to intelligence. On the other hand, we also have genetic studies where in genome-wide association studies, people study a large cohort of individuals and try to identify what is the variability in genetic factors and genetic variants that associate with intelligence. So they provide this genetic variability, but also insights into which genes associate with intelligence. But in the end, there is a big gap between those strategies. So we know that brain doesn't function like only on the level of the whole brain. Brain consists of areas, different functional areas, and networks that combine the activity for intelligence. The areas consist of networks, individual neurons, and finally individual neurons express genes that will affect their function and structure. So there is a whole to say on this multi-level, multi-scale about how our biology underlies intelligence. And in our lab, we are actually interested in all those scales. So the question that we ask is how genes, neurons, and networks support human cognition. And of course, if you want to have all this data in the same individual, it's quite challenging. So we came up with this approach that we've been applying already for almost 15 years in our lab, where we work with patients who are treated for tumor on epilepsy. And usually these patients have a lesion or epileptic focus somewhere deep in the brain, in the hippocampus or amygdala, or a tumor, again, deeper in the brain. And in order to get to this deeper lying focus of the disease, the neurosurgeon has to resect a piece of tissue that is not pathological, just the excess tissue, and that we can actually take into the lab. We physically go to the lab, to the operation room. So these are two PhD students from our team going in the operation room to be there as fast as possible, and as the neurosurgeon resects this precious piece of tissue, we can take it in our specialized solutions so that we keep this tissue intact, neurons alive in their intact networks, and then we can take this tissue in the lab. This is how a typical piece of human cortex will look like. And then we slice this tissue so that we have those individual slices to look at the structure of the tissue, and then we can use electrophysiological equipment to zoom in on not only the structure of the cells, but also on the electrical activity. So this is, for those of you who are not familiar with cell electrophysiology, this is how this typical rig would look like. And the slices here under the objective and the microscope, and you can see those electrodes, the stages with electrodes directed to the cell. And on the screen we can see the readout from what we see from the microscope, the electrodes, and then we have this electrical activity in the cell that we can monitor the health of the cell and also what kind of electrical activity the cell can produce. So in this way, we can study the spontaneous activity of the cells. We know they are alive, that they are online, they are healthy. And we can also look at simulated activity. We can induce activity in the cell and see how the cell will react with an output. So it gives quite a localized and a very high resolution gives high temporal resolution and high spatial resolution to study the electrical activity in neurons. So in this way, this is how this cell will look like on our screen while we record. So you can see a cell body with an electrode attached to it, and then all the dendrites you cannot see in this type of image, but then we can fill the cell with biosighting, with dye, and then after the experiment we can perform staining so that we can see the full morphology of this cell, or small network of cells, and then also reconstruct this morphology, and then also study how dendritic structure is related to, for example, intelligence. So in this way, as I mentioned, we've been doing this already for some time. We've collected quite a comprehensive data set from many subjects where we would have their MRI scans, MEG activity, and EEG activity. Because we have the tissue in the lab, we can also look at fine architecture of this tissue at the level of the neurons, but also layers. We can perform electrophysiology to look at physiological activity of single cells. And then recently we started also retracting the nucleus from those cells, the nucleus that contains own RNA, so all expressed genes in this individual cell. So in this way we can use modern sequencing technology to also look at the expression levels of genes in every single cell. And then to look how different cells express which genes and to which levels. So let me show you some insights that we collected from working with this type of data set with these patients. So first of all, as I mentioned, there is already a lot to be said about the structure and function of different brain areas in healthy humans. And also Roberto contributed to this research. So also one of your papers looks at the relationship between activity and structure of different areas of the brain and intelligence. And this is kind of an overview that we also used in our first studies. Comes from a Deary review paper, where what you see here is actually quite a distributed number of areas. Again, as Kirsten mentioned, a lot of areas are involved in intelligence. And in this particular review, you see the areas that relate in their cortical thickness to intelligence. And what I want you to pay attention to is the differences between left and right hemisphere. So it's only structure, it's not function, it's only structural thickness of the cortex. But you can see that left is different than right. And what is also kind of interesting to us is that if you look at the left hemisphere and have some knowledge of neuroscience, you would see that actually these areas correspond very much to language areas. So Broca area somewhere here, Wernicke area, and also the semantic area of our brain and the temporal lobe. So this area, of course, we know that IQ tests also relate a lot to verbal intelligence. So one big component of IQ test is verbal intelligence. So in the rest of my talk, I will show you a lot of data that looks at verbal intelligence, but I want you to keep in mind that everything I will show you is actually true also for general intelligence, because verbal intelligence is so much correlated with general intelligence. Actually, the data seems very, very similar for those two different measures of intelligence. So yeah, we saw this type of interesting research, and we realized that when we get the tissue from the surgery, it almost always, because of the surgical procedure, it almost always comes from this area 21, Brodmann area 21. So we thought, we always were thinking, how can we understand what this area does? And then we realized, well, it does have a function, but this function is very general. So it's related to this really general intelligence or verbal intelligence, and we should actually test it in our data. So the first question we ask is, in our small data set of patients, can we actually see the same what people observed in healthy participants, that cortical thickness relates to IQ scores? So for this we use the MRI scans from those patients, and we analyze the cortical thickness in the whole brain. And then, because we were interested in the temporal lobe, we only focused on the cortical thickness, the average cortical thickness within the whole temporal lobe. And then we asked whether IQ score correlates with this cortical thickness in the temporal lobe. And indeed, we did see a modest but significant correlation. So the subjects with higher IQ scores also had larger cortical thickness in this area. And what is actually quite encouraging to us was that if you look at meta studies of MRI-based gray matter volume relationship to IQ scores, you see very similar correlation coefficients, and actually also quite a lot of variability in the data set. So that was for us encouraging, but we digged further because, of course, we have also access to more precise measurements of cortical thickness from our histology. And here we were asking what kind of, can we see any relationship between specific layers of cortex and, so not the whole cortical thickness, but specific layers and IQ. And our hypothesis was that if we inform ourselves from the evolutionary studies, since we are so much interested in evolution, we know that human cortex is thicker compared to other species, but it's especially layers 2 and 3 that are expanded mostly. And the reason for this could be that these layers contain neurons that send long-range projections to other association areas. And in the human cortex, of course, association cortex is very much increased, so there is more space for these long-range projecting neurons. So that's why we started to identify different layers and correlating the thickness of these layers in our data. And what you can see already maybe from this example is that if you look at the whole cortex, you see that indeed whole cortex in a high IQ subject is thicker, but this thickness is mostly due to layer 2-3 thickness. All other layers remain more or less similar. And that was also exactly what our data showed. So here I'm showing you our data set split in two halves on high IQ and or high verbal IQ and low verbal IQ subjects. So high IQ subjects are shown in red. And we did this analysis for all layers separately for left and right hemisphere, because as I showed you, left hemisphere has very strong correlations with IQ. And what we found was that only in the left hemisphere and only layers 2-3 we see this difference between high and low IQ subjects in cortical thickness, and so in layer 2 and 3 thickness. And this is, of course, kind of a split between two groups, but we could also look at the level of individual subjects, and we saw actually very, very strong correlation. That it's very difficult to, in all my research, I've never seen any type of, this type of strong and significant correlation for a data set. So, you can see the difference between what I've shown you for cortical thickness from MRI measurements that was very variable, but here the relationship was much stronger. So we concluded from this that, especially in the left hemisphere and in layers 2-3, we see this relationship between IQ and cortical thickness. And of course, there can be different scenarios why this cortical thickness is increased. So one scenario that you can think of is that there are more neurons, that these layers contain more neurons and more connectivity. And another scenario would be that the neurons themselves are different. So which one do you think is the correct answer? The first one, more neurons, or the second one, the different? Second. Both. First. Okay. So many people say second, and indeed, you're right. It's the second scenario. So here I'm showing you just an example of two Is it a question?
Yes.
Yes?
On how many participants are these results based on? So when you go one slide back, there were 16 points
Yeah.
also. Is it?
Yes, it is indeed a small data set. And with every, okay, so we collected all this data from the same, let's say, cohort of patients. So totally, I think there were 79 patients, so a small data set. But of course, for each measurement, there'll be some patient that we didn't have, that the slices were not good enough. So we were losing some patients for some data sets. So it's very, very much undersampled. And again, that's why it's so surprising that we see such strong correlations. And I must say, why I think it worked is because we had this very large range of IQ scores in our patient data set. Usually when you work with healthy undergraduate students, it's more or less the same data set. Here we had a range of 60 to 135 IQ.
So do you have a chance to replicate it?
No. No, not yet.
Okay.
Yes.
This is very interesting finding from the electrophysiological point of view, because typically in those areas is where it's supposed to come the gamma band. I mean the alpha and the beta goes from lower layers, but the gamma comes from there. But never, I mean, because as you commented in your talk, it's very difficult to see it in EEG. But then we should focus more on this gamma band to see differences between high IQ.
Yeah,
and
so
IQ.
we're trying to do this now, analyzing MEG signal.
Yeah.
We actually don't find much in gamma band.
Okay.
We were hoping for gamma band or beta again, because it's also connected to layers 2 and 3. But it's actually in the alpha that we find differences. But I would be careful still not to go in there, because it's very preliminary and unpublished data.
And replicate.
And so the point is, it's very difficult to even collect human tissue from these patients. And I think at the moment we are a handful of labs that even work with human tissue, and especially with IQ tests and other measurements. So it's very precious data set, and we cannot. This was already collected in I think within 10 years to collect all the data. So there must be ways to do it more faster and more organized, especially with collaboration with other labs. We're trying to set it up now, but it takes time. So I hope that we can at least replicate this in other data sets and go further. But then coming back to the structure of those layers 2 and 3, so in the example that I'm showing you, these are two examples of two different patients, one with thicker layers 2, 3 and another with thinner layers 2, 3. We didn't, from this data set, from this data set, we didn't have IQ scores. We only had the structural thickness of these layers, so we don't know whether this really relates to IQs, but we know that if we look at the thicker or the relationship between thickness of layers 2 and 3 and the structure of this tissue, so the density, neuronal density, or the cell body area, we see very strong correlations. So if when the cortical slices have thicker layers 2 and 3, usually it goes together with lower densities, so not more neurons, but this actually the same number, more or less the same number of neurons, but at lower densities, and also the cell body size increases. So these are bigger neurons in a larger volume, but not more neurons. And then the question is, what is this volume for? Because what you see here, this white background is of course occupied by glial cells that we are not showing this staining, and connectivity. So we know from animal studies that 95% of this volume is occupied by dendrites and axons. So it's really densely packed with connectivity. So for this, we turn to this more fine structure of these neurons and to the full 3D reconstructions of the dendritic trees of these neurons. So now we are not looking at cell bodies, but the full structure of these neurons. And again, as you can appreciate from this example, we saw that indeed there is a quite strong correlation between IQ and the structure of these neurons. So the higher the IQ scores, the bigger the total dendritic length, so the size of the whole dendritic tree in these cells. So the squares show mean data per patient, and we had, for many patients, we had several cells that we recorded from, so this is now averaged. And also the complexity increases with IQ. So people with high IQ scores have larger neurons with larger dendritic trees, and these dendritic trees are also more complex. And you can imagine that these dendritic trees are there not for nothing, so they receive a lot of inputs from other neighboring neurons or long-range projection from other areas, so they integrate a lot of information. And yes, for sure.
How do you measure the complexity? How do you define it?
Yes, we define it by the number of branches of the dendritic tree. So there are, if you can see here, there are nodes where the dendrite branches. So we quantify how many of those nodes we can find. Mm-hmm. There's another question.
What about dendritic spines?
Uh-huh.
Do they have to do with
Yes, that's an excellent question that we discussed during dinner yesterday. So in our research, this research, so when we fill the cell with biosighting, it doesn't give a complete fill. So we don't have enough resolution to really look at all the spines. So we do see spines, and we tried to quantify this. And when we did this, we didn't find any correlation. But we never published that because we think that the method we use is not sufficient. So I just heard yesterday that Javier de Felipe, another group working with human neurons, are trying to do this, and I would be very curious to see how this what kind of results they will collect. Because my intuition is that there must be some relationship, but the thing is that the dendrite, so there is, of course, constraints on energy and volume in the human cortex. So we know that these dendrites are packed with spines. So there's actually not even enough space to grow more spines, because that's already all the space is taken. So my hunch will be that it's not only the density, it's these dendrites are longer, and because of that elongation of dendrites, there is more physical space for more spines. But if there is no elongation, it's probably all this volume is already occupied. But that I would be very curious what the results will be. Okay, so I was yes. So I mentioned that we saw many of those correlations in the left hemisphere. So we also split now already small data set in left and the right hemisphere for total dendritic length. And we actually, to our surprise, we found that here also we find correlations in the left hemisphere, but nothing in the right hemisphere. And actually, if you see from these graphs, on average, total dendritic length is longer in the right hemisphere, but it does not correlate with IQ. It could be undersampling, but it could also be that indeed left hemisphere is kind of dominant hemisphere, more involved in language, more involved also in the IQ tests, that it plays that these neurons play a bigger role in this specific test. Okay, and then of course these big dendrites receive a lot of inputs, and they process a lot of information. So we also were wondering, what about physiological properties of these neurons? How do they react to inputs that these cells will receive? And of course, the bigger the dendrites, the more inputs the cell will receive. And one way to look at this is looking at the output of the neuron and the action potentials. So we recorded action potentials at different frequencies in these neurons. So we will apply currents, and then the cell usually will respond with action potential trains. And then we analyzed the shape of action potentials in all those neurons from people with high and low IQ scores. And what we found is that actually in low IQ patients, the shape of action potential is slower. And that was also surprising to us, because we read in neuroscience books that action potential is an all or none event. It's either you have action potentials or you don't. But actually it's not completely true. We also know from rodent studies there is variation in action potential speed. And this variation does matter a lot for computational properties of these neurons. So just to show you first the quantifications. So we again split our data in the left and the right hemisphere, and we looked at action potential speed at the first action potential of the train, but also action potentials at different frequencies. So what we found is that the higher the frequencies, the more slowing down we saw in neurons from patients with low IQ scores. But high IQ score patients had neurons that could keep up with this speed of action potential and maintained action potential shape. And this is the same data shown for this 20 to 40 hertz range. So again, these frequencies do not mean that the cell fires at 40 hertz. It means that action potential instantaneous frequency, so how fast it happens compared to the previous action potential, is at this higher range. So sometimes it will be just a burst of 2 or 3 action potentials, but if it's in this range, we would count this instantaneous frequency. And again, we see the correlation for individual patients. So the higher the verbal or general IQ, the faster action potential rise speed. And again, in the right hemisphere, we didn't see any correlation. So this holds true only for the left hemisphere. And maybe for people interested in more computational aspects, we also had a model where we were interested how this action potential shape affects and the size of the dendritic tree affects input-output function of the neurons. So this can be, there's one way to study this is to model the inputs to the cell at different frequencies, so as a sinusoidal wave that you can range from 1 hertz to 1000 hertz. Then you add up some noise to the signal. And what the cell will do, we actually performed this experiment also in human neurons, but this is just an example to show you. So what the cell will do, it will spike sometimes in phase with this oscillation. It will never spike with each cycle, but it will time its action potential to this sinusoid wave. And then when you increase the frequency of input, it becomes more and more difficult for the cell to time action potential to the sinusoid. So it will lose its reliability. So we can quantify this type of reliability or encoding of information in a response magnitude for each cell. And we actually modeled all different dendritic lengths for each cell, because we had this realistic morphologies. And we modeled how this dendritic tree can affect response magnitude at different frequencies. And you can see there is a lot of variation, but the smaller neurons cut off at lower frequencies than bigger neurons. So bigger neurons are able to respond reliably to higher frequencies compared to smaller neurons. And actually this cutoff frequency, so when there is this cutoff, when the neuron cannot sustain this, can reliably encode inputs, this cutoff frequency correlates with total dendritic tree. And actually this type of data we can also based on this type of data, we can compute information rate of each neuron. And this cutoff frequency would also mean that there is more information that the neuron can encode. And so this depends on the total dendritic tree, but also on the how fast action potential is. So this means that neurons from subjects with high IQ scores, because of these structural and functional properties, are able to process more information in a certain amount of time. Okay, so just to have a small, slow down a bit and recap what I've been talking to you about. So I told you that in our research we find that there is a relationship between cortical thickness in temporal lobe and IQ scores. We also find that this relationship is much stronger for layers 2 and 3, so they are enlarged in people with higher IQ scores. This is probably due to expansion in the total dendritic trees of these neurons. And these changes come together with physiological adaptations where neurons are able to fire fast action potentials and respond faster to fast changing inputs, and in this way encode more information. So this is so far for brain areas, networks, and neurons, but how about genes? So I want to switch gears here a bit and to talk to you about now our attempts to also connect genetic studies to what we found on the neuronal level. So I don't know how you are familiar with GWAS studies, but just a small reminder what actually GWAS studies are able to do. So GWAS studies, genome-wide association studies, study large cohorts of people with, of course, genetic variability. They look at specific parts of the genome where there is variation in the genome in the single nucleotides. So they're actually mapping this single nucleotide polymorphisms. So places in the genome where there is the differences, there are polymorphisms between people. And you can imagine that if you start correlating these frequencies of single nucleotide polymorphism in different people to specific traits, then you would expect that, for example, if certain polymorphism like A will occur in 90% of people with high IQ scores, then you would expect that there is some relationship of this polymorphism to IQ scores. So basically, these GWAS studies map a SNP association expressed in p-values of the correlations for each SNP at many places in the genome at different chromosomes. And then once there is statistical significance, so above certain value, you can consider that those SNPs associate with a specific trait. And as in this example, that these SNPs would associate with IQ scores. And once you know which SNPs and where associate with IQ scores, you can actually map those SNPs to specific genes, because it's known where in genome these SNPs are, whether they are close to specific genes or in the gene. In this way, you can map which genes associate or are candidate genes that could associate with intelligence. So fortunately for us, we didn't have to do this time-consuming research. It was already done by other groups, one actually at our institute. And we focused on two studies. So one from our institute that identified about 1,000, so 936 genes associated with IQ. And another study that used a proxy of IQ educational attainment, so how many years a subject has spent in education. So we focused on these gene sets and wanted to explore a bit more what these genes, how these genes relate to cells. But since we're also interested in evolution, we also took another gene set, a gene set that comes from comparative genomics and that identified a gene set of so-called HAR genes, so human accelerated regions. So these genes are genes that show accelerated evolution in the human lineage compared to primates, especially to our closest cousins, chimpanzees. So the idea behind this is that if there is accelerated evolution in those genes, they probably matter something for our human brain evolution. So we were also interested in this gene set. And the problem usually with genetic studies is that they look, of course, in the whole genome. They look at associations of specific genes with traits, but they usually what they show is what you see in the statistics of the whole genome, that most of the SNPs or genetic variation will be intergenic or intronic, so in the genes that are not coded to proteins. So only about 2% of all this variation will be in protein coding genes. So we don't know what the rest of this variation does, and there is only a very small percentage in these protein coding genes. And then if we focus on those protein coding genes, most of them, if we look at studies of intelligence, most of them are involved in very early processes of brain maturation. So in neurogenesis, probably that happens during embryonic development, or neuronal differentiation, again, very early processes. So it's not that informative if we are looking at adult individuals and their IQ scores. But there is a minority of the genes that are involved in synapse structural function. These genes actually are much more interesting. And again, problem is that all those genes show very small effect size for each gene. So you really have to take polygenic scores, combine information from many genes to get any type of meaningful result. And we don't know whether they are expressed in the brain, because they can be expressed, of course, in any tissue of the body. We don't know where in the brain they express, in which neurons. And we don't know whether they are expressed in the adult or developing brain. And for this, we have a very powerful tool nowadays, transcriptomics, that can actually answer those questions. So transcriptomics does not look at the whole genome. Transcriptomics looks only on RNA in individual cells. So RNA will give a readout of expressed genes, only genes that are active in this particular cell. And we started a collaboration with the Allen Institute for Brain Science in Seattle that is they're kind of an expert on RNA sequencing tools. It has a big database on the RNA-seq data for human brain. But we combine this approach with our experiments. So instead of looking at all nuclei in the tissue, we actually conduct our physiological experiment. We do our stainings for morphology, so we have the structural and functional readout from these cells. But as I mentioned, in the end of the experiment, we can retract the nucleus with RNA and then use the sequencing methodology to get the readout of all expressed genes in this particular cell. So in this way, we can combine morphology, physiology, and transcriptomics in the same neurons. And by using this approach, together with the Allen Institute, we uncovered that there are several types of pyramidal neurons in layer 2-3, so the layers that we're interested in. And if we compare those types to mouse types, we see that some types are homologous to mouse, so they're very similar. But there are also types that do not have equivalents in the mouse brain, so they are kind of human specialized types. Although, to really make a conclusion about human specialization, we have to compare it to more species than only mouse. So in this way, we have the readout, as you see here, for morphology, physiology, and also the cell type, and all the genes expressed in the same cell. So, weaponed with this new technology, we decided to ask the questions. If we take those gene sets that are already published for IQ, for human brain evolution, can we actually say something whether these genes associated with human brain evolution or intelligence expressed in specific brain areas, that like association brain areas that are involved in cognition, are they expressed in adult neurons, and are they expressed in specific neuron types? We can also, because we have the readout of dendritic morphology and physiology, we can also ask the question whether these expression levels of these genes relates to structural and functional properties of neurons. And finally, we can also look at what is the function of these genes and which genes are overexpressed in these neurons. So for this, we focused on three gene sets, which I mentioned: the educational attainment set that was published, IQ set, and HAR, so human accelerated region set. And we asked whether these genes, if we take average expression within the gene set, of all those genes, just calculate one value, average gene expression, whether they are overrepresented, overexpressed in specific areas of the brain. And we found that there is a slight increase in association areas of the brain, and specifically in area 21, so middle temporal gyrus in the human cortex, so exactly the area where we receive our tissue from. And then the next question we asked was, how about the specific classes of cells, glutamatergic, GABAergic, or glial cells, whether there are differences in expression of these gene sets. And what we found was that especially in glutamatergic neurons, so pyramidal neurons of the brain, there is high expression of these genes, so average gene expression of IQ data set, but very similar results also for educational attainment and hard genes. So you can see here already, we see kind of a crosstalk between the genes that are involved in human cognitive ability and brain evolution. So here we used RNA sequencing database of Adult Brain Institute. So these are really like thousands and thousands of nuclei. But this data does not have morphology and physiology. So after confirming this, we turn to our patch-seq data, where we have all the modalities, and we focus on these 5 types. Actually, one we split in 2 because it's a very variable type, so deep frame 3 and layer 2 frame. And we looked within these cell types, what is the expression level of IQ genes and other gene sets. But before I mention this, I want to show you that what is important is that there is this variation in morphology and physiology within this, between these cell types, and especially human-specific types that do not have analogs in mouse. They show this very branchy morphology, so a lot of dendrites, and also the same types have fast action potentials. If you look at these arrows for purple and blue, they have fast action potentials than other neuron types. So before I was talking about pyramidal neurons as one population and comparing IQ subjects for those features. But of course there is this diversity of neuronal types in human cortex. So it's not that all these neurons are faster and larger. So it's probably especially these large and fast types that are either overrepresented or have enhanced neuronal properties compared to other neurons. But this diversity is probably preserved. So now if we look at the quantify the dendritic length or action potential speed in these neuron types, we see that especially these human specialized types, so CARM3 and FREM3 types show more expanded dendritic morphology and also higher rise speeds. So what about the expression levels of IQ genes and other gene sets? Here again we find the highest expression of genes associated with IQ in these cell types, but also in the educational attainment gene set and in a hard gene set. So there is an overlapping pattern of expression in this layer 3 large and fast pyramidal neurons. And finally, we wanted to know which genes actually from these rich gene sets correlate most with the neuronal phenotype. So for this, we performed actually quite a simple analysis, just a correlation analysis, where we arranged all neurons according to their neuronal features. So, for example, total dendritic length from small to large cells. And then we looked for each gene, for those thousands of genes, what is their gene expression level that is shown here in color. So some cells will have higher gene expression levels, some will have lower gene expression levels. And then we were interested, which genes actually show highest correlations. So for this we ranked those genes according to either positive correlations that you see here, so from small to large expression level, or negative correlations where there is large expression levels in smaller cells and then lower in larger cells. So this is an example of how this, the results of this analysis looked like. So here you can see example for IQ gene set and correlations with total dendritic length. So neurons are arranged in total dendritic length in columns. And then we selected the most correlating genes, positive correlations and negative correlations separately. Of course, compensating for multiple comparisons with FDR of 5%. So we repeated this for total dendritic length and action potential rise speed for all gene sets to identify only genes that correlate. And then if we look how these genes, only correlating genes, are expressed in these types, you can see that the differences become much bigger between cell types. And again, these human specialized types stand out. So they show the highest expression of these correlating genes. And another thing that we found out is that when we started with this analysis and we looked at those gene sets, there's actually overlap in which genes are involved in which gene set. So there is always this overlap between all three. And if we looked at educational attainment genes and IQ genes, so which part of those gene sets is also involved in the brain evolution, so in also proportion of HAR gene set, we see this 19-14%, so modest percent, but still quite interesting that there is this overlap. But then again, if we only look at correlating genes, then this overlap becomes much, much bigger. So there's, of course, smaller gene sets now because we only use the correlating genes, but you can see that the proportion of HAR in these gene sets is almost 50%. So it signifies that the processes that are involved in human brain evolution are also similar to those that explain variability between people in IQ. So it means that also evolutionary processes that we study and these genetic processes are important also for cognitive ability, of human cognitive ability. So in the final question, before I will finish, what is actually biological function of these correlating genes? So far I've been just showing you like clusters of genes, but of course these are individual genes that have a certain function. So to answer this question, we performed overrepresentation analysis that compares the frequency of genes involved in certain biological processes compared to background frequencies of all genes and all biological processes. So it means that if in a gene set there is a group of genes that is involved in certain process, you can actually statistically compare this overrepresented biological processes to background ratios and use statistics to inform you whether these genes are overrepresented in specific processes. So what you show here, again, ranking of most overrepresented processes to least. And here you show the p-values and individual genes marked with squares for those genes that are involved in specific processes. But what I want you to pay attention to, actually, it's a pity that the contrast is much worse than on my laptop. But there is a highlighting of in a light green for those processes that are involved in synapse. And actually, it's almost exclusively all those processes that are overrepresented are involved in synaptic function. So you can read this glutamatergic synapse, synaptic membrane. So everything is related to structural function of synapse, axonal growth. So almost nothing with a high-level function like transcriptomic factors with a known function. Although it's a minority of the genes in those GWAS studies that are related to synapse activity, in this, if we come down to this small identified gene set that relates to function of neurons, these processes become much more overrepresented. And what we're actually doing now is we are studying the most interesting genes from this list. What is their function in the synapse and how are they increasing the computational power of the neurons, and in this way of networks and finally intelligence. So one of the targets that we are interested in is in glutamatergic receptor 3, but also in this list you can see a lot of subunits of voltage-gated receptors, ion channels, and etc. Okay, so I came to my last, almost last slide, but the summary slide. So what I have been talking to you about is differences on the cellular and network level between people with high and low IQ scores. So to summarize this, of course, you're all very population of people with high IQ scores. So I will be very confident talking about you in my summary. So how is your brain different from other people? Is first of all, you have different cortical organization. So you have thicker temporal cortex with lower neuron density and expanded volume, especially in layers 2 and 3. You have, if you look at the individual neurons, your neurons probably have larger dendritic length and more complexity, and they are able to produce fast action potentials in pyramidal neurons, and this translates into computationally in a better way to encode information. And then if we look in these specific large and fast neurons, pyramidal neurons, and we look at their gene expression of genes that associate with cognitive abilities in humans, we see that expression patterns of these genes, so for brain evolution and cognition, converge in these layer 3 pyramidal neurons. So all data sets actually show similar things, enrichment of these genes in layer 3 neurons. And also genes that associate with human cognition correlate with neuronal properties. They have mostly synaptic functions, and half of them are part of the gene set that is also implicated in human brain evolution. So like in the overall take-home message would be that if we look at the processes that are involved in human brain evolution and specialized properties of human neurons, these are actually very similar to those properties that are relevant for human cognition and explain variability in individuals in their cognitive ability. So just to finish off with acknowledgement and the photo of our lab and all the people who have contributed to this research. And thank you very much for your attention. So I'm open for questions.
Thank you very much for your very exciting talk, Natalia. And she's now open for questions.
First, thank you very much. Thank you very much for this great talk, very clear, and I love your slides. I have several questions, to be honest. So the main thing that I had in my head is, what do you expect? You would have I'm sure you don't have. What is the age range of the participants?
So between 20 and 65. Mm-hmm.
Okay. So what would you expect? How this develops these neural characteristics? So you demonstrate the link to genes, but the link is not one-to-one. So what role can we conclude something, or what would we expect? What role the environment plays with switching on the genes or epigenetics? And the other, maybe one question is about sex differences, especially concerning the hemispheres.
Yeah. So to answer your last question, we don't find any sex differences. So for all those parameters, we compared males with females. Nothing stood out. And to answer your first question, the developmental, how developmental things, how this will all add up. So I think the strength of our study that we really looked at the adult population, adult neurons, where we can measure IQ just a couple of weeks before the surgery, and then genes and neuronal function, very close in time to IQ measurements. So it's just a readout of that moment of adult neurons. But of course, there is a lot of for example, from genetic perspective, what we find is most of those correlating genes are protein coding, they're involved in synaptic function. So it's really something that already influences synapse. But a lot of those genes are high-level regulatory factors, transcription factors. So they would be much more involved during development. So my guess would be there is a lot of room for epigenetic, but we don't pick it out because we are looking at the already crystallized adult function. And also for dendritic trees, there is also a lot of room for growth and neuroplasticity. So we know that dendrites grow or shrink depending on age or life conditions. So again here, something for sure would change during the course of life. So a lot of room for environmental factors.
It would be great to see these effects on the neuronal level with action potentials and so on at different stages during childhood.
Yeah. There's a question. Yeah.
First, thank you for your talk. It was really, really interesting. I have 4 questions.
Okay.
First of all, if you've done any kind of like longitudinal studies, longitudinal, like if you've evaluated these people, if you have any information of how these people have been during their childhood, if you have evaluated any person since you've been doing it for like 10 years, if you have had the chance of evaluate anyone during their childhood or younger, and then evaluate them later and see any difference because of plasticity.
Yeah. So no, because these are surgery subjects. So they undergo surgery, and that's the time point where we can take the tissue. So they don't come to surgery, fortunately for them, second time. But what we know is that after the surgery, there is still a neuropsychological evaluation, and these people usually do improve in their IQ scores, except for high intelligent people and left hemispheric resection. So exactly where we find our correlations, they usually lose in their ability, and why they improve and is probably also because of a redundancy of the whole network that Kirsten was mentioning. So even if a big chunk of the brain is taken out, people are seizure-free after that. They don't take a medication anymore, so that's the improvement. And the rest of the brain takes over the function of the temporal lobe. So intelligence is not only, again, this shows that intelligence is not only the small piece of the brain that we take out and then the person loses intelligence. No, it's the whole brain, and we just have a representative area.
Okay. Okay, thank you. Then the second question was if you have had the opportunity as well to, or had any data from people with language, problems, like aphasias or stuff like that, and see, because it correlates so much with language, if you've seen differences in their thickness of the temporal lobe or Wernicke or Broca areas, if you've seen any difference there, because they also communicate, but they communicate at another level. So if you've seen any difference there.
No, we haven't done that, but I'm sure there is a lot of studies in healthy people who try to look at network or like area, like Broca area function and language disability. So I wouldn't be confident in giving you an answer on that, but we were not able to do this because we're only working with the small area of the brain that is not the Broca area, for example. So usually these really critical areas are spared during surgery because otherwise people would lose their language function completely.
Okay, thank you. Then the 2 last questions. Because you've said you also compare in a kind of like evolutionary way, and you've focused mainly on primates because it's been studied as well in higher birds, let's say. which are also the singing birds. So they have kind of like a language really developed part of the brain, and they also correlate with a big number of neurons. So you said number of neurons, a lower number of neurons and a higher volume correlates with higher intelligence, also with higher language intelligence. But these birds, passer and singing birds, they have a big, like they have among vertebrates the largest number, the largest density of neurons, and they also have, and they also sing, so they also communicate a lot. So if you have done any evolutionary study, or if you've read any evolutionary parts implying birds as well?
Yeah, no, we haven't been studying birds, so no chance to compare that. But for the number of neurons, so what we did is comparing within this like 2D surface volume, the number of neurons. But of course, the brain is three-dimensional. So I could imagine that people with high IQ scores, they have also more like 3D surface that would have also more neurons. So it's not, so like if we really do those cuts and we look at the volume, yeah, we see that the number of neurons doesn't change, but most probably there is more volume, the gray matter volume, and also more neurons in absolute numbers in people with high IQ scores. It's just that we couldn't compare that. And I think, yeah, what we add is not only, because it's kind of, it's like a known fact that number of neurons would add to computational power of the brain. And we know that human brain also have more neurons than other primates. But I think what is also important to realize that it's not only the number of neurons, but also the properties of individual neurons that correlate, like dendritic trees and complexity.
Okay, thank you. And yes, and the last question. If you found any homologue genes to intelligence, because also it's been described like animals that have higher intelligence than others, mainly like high vertebrates, if you can call them like that. If you found like any homologue of those genes that you found in humans, in other animals, if you've done like that kind of evolutionary study.
So no, we haven't been looking at homologue genes in the animals. But what we are planning to do is to take some of the targets that we identified with our analysis and to compare also across species. What do these genes or their products, like receptors, for example, what do they do? How they compare across species? What are the differences in the function of those genes? But I think it's difficult because even so you can find a homologue of a certain receptor. But there's no study that does kind of a GWAS-related study in animals. So then there's nothing to compare because we just don't know.
Okay. And in these genes that you found in humans, the single nucleus, the SNPs, they are just like the higher IQ the people have. They are just expressed, or they are more expressed. Like
they're more expressed. Yeah, they will look at the levels of expression. So it's not that the gene, all of those genes are expressed, almost all of them, but the levels of expression are different.
Okay, thank you. Thank you.
Hello. You mentioned that people with higher IQ have larger neurons, and my question is, why are they larger? Like, they just happen to be bigger than other people, or there's a reason behind it?
Yeah, there's certainly a reason behind that. So larger neurons means more dendrites, so more space for integrating inputs. More complex dendrites mean actually that computational operations can be localized in different branches and performed separately. So in this way, one neuron can actually act as a network of neurons. So each branch will be kind of a separate node. So it increases the complexity of information this neuron can process. And our physiological measurements will also show that action potential speed and dendritic length directly translates into higher information rate. So how inputs are translated into outputs.
Okay, thank you.
Okay, thank you so much for the talk. I'm sorry for my voice. So, I don't know if it makes sense, but it's known that as our brain develops, we lose gray matter density, and also there is a change in the excitatory and inhibitory balance. So we lose excitatory, especially prefrontal excitatory synapses, as we gain inhibitory synapses. So is that the reason why Adam showed us that our best years are between 14 and 16?
So I'm not sure about our best years between 14 and 16. I would not subscribe to this. But so what we know from developmental studies, also studies of intelligence, that it's actually a very complex thing. So now we are looking at, again, in a time point in already adult development. But if we look at the developing brain, there is this dynamic of the development. And you're right, there is pruning of synapses, shrinkage of cortical gray matter, but there's reason for that because there's selection of relevant inputs. But what we know is that the development in children with high IQ scores is usually more protracted than in children with low IQ scores. So they actually need longer to mature, and sometimes at the same age they will perform worse because their brain is not mature yet compared to other children. Am I saying correct things? Again, that's not my area of expertise. So if we look at development, there are more factors that we have to take into account. So it's not just branch neurons will relate to high IQ scores. It's this temporal dynamics that we need to face.
Thank you.
Yeah.
Thank you. First, thank you for the talk because it was really easy to follow and the slides were really good actually. And my question is more like a bit random. It's just about like the future of this type of research because I'm curious to know because, for example, in the past we couldn't be able to detect certain anomalies in the genes and stuff. So I don't want to sound like conspiranoic, but I don't know if this type of for the future for this research, would it be, do you think is in the future path to be like a type of change or like modify gene to express or express more intelligence sets of genes?
Yeah, it's related to the pill, the intelligence pill question, right?
Because there's but
even worse, even cutting in the genes.
We are moving closer.
I think we are safe there because there are thousands of genes that are related to intelligence, very small individual effects. So if we had to change our genes, that would be, we first have to target specific cells, change the genes there, and also change thousands of genes. That would not be ever doable. And even then, if we would do that, I know that I think for like latest US studies, it's only just a bit more than 20% of variants that all those genes together explain. So again, there's 80% of variants of differences in intelligence that is not explained by genetics, or at least this sets of genes. So there's a lot of room to still, we are safe from genetic interference.
Not like in the movies, no?
Yeah, not like in the movies.
Thank you.
Yeah. Which gives hope to us that there is, we can still maybe change our intelligence and change our epigenetics. Yeah.
Natalia, thank you very much for your great talk as well. The idea of having the correlation between the how large are the dendrites and intelligence, I don't understand why. I mean, because of what is the potential mechanism that are facilitating speed processing. So it's I'm guessing it's because you have a higher delay in signal, so you allow the neuron to recover to have another potential then much faster, or what are your hypotheses?
Is your question about the relation between dendritic length and action potential speed? So how they two relate? Yeah. So as I mentioned, bigger dendrites would mean more inputs. So we know that one cell, a human cell, can have more than 10,000 synapses, and you can imagine that in vivo, in a real brain. This cell will be bombarded constantly by all kinds of inputs. So for the cell to process those inputs in a temporal dimension, it needs to react fast to relevant inputs and process signal from noise. So action potential speed is actually directly related with large dendrites. We know this from computational studies, but also from animal research. So usually neurons with large dendrites would have fast action potentials. And the reason for that is also this computational ability. So more inputs at the same time would be resolved much better with fast action potential. So the cell reacts faster to each individual input. So it's not let's say, how fast the cell needs to fire in terms of how frequent it needs to fire action potentials, but it's the ability to resolve all those multiple inputs within a short amount of time.
Okay. Okay. I was just thinking that the delay between the input and the time it takes the signal to go into the soma, that could help a lot as well, because this length that will delay the signal and then allows the dendron to process another stimuli. So the larger, the better.
Yes, but it also delays information. So
allows to process more complex inputs as well.
Right. So there is also a phenomenon of dendritic filtering. So these large dendrites will also actually make the signal will lose strength along the dendrites because of that. So it's there are also mechanisms in human neurons to counteract this dendritic filtering. But I have been talking about this.
Good. Very good.
Yeah. So there's more going on than only dendrites and action potential speed. Yes. There's a question.
No, thank you for your talk. And it's only a suggestion. From an electrical point of view, the thicker the conductor, the better the response to high frequencies. So the losses are low at high frequencies. And even if the thicker the conductor is, the losses in any frequency is lower. So it's a best conductor, a thicker dendrite or a thicker axon. Yeah. Of course, the other problem is the length. So if the length is higher, the signal takes, at least in theory, takes more time to transmit.
Yeah, that's absolutely true. There are also studies that show, like of our collaborating lab, that show that thicker dendrite is the most important factor for signal speed. From dendrite to soma. So indeed, not to lose the information. But there are also separate conductances, so specific channels that are more involved in human neurons that can also accelerate signal from dendrites to soma. So, but thicker dendrite by itself already means a lot, improves a lot. I will sit down.
You see that we are now entering to the final stage of this course. Yeah. It is the round table, so make questions for everyone, okay? So
maybe with the other speakers can also join, and we can.
Or maybe what about making a 5-minute break to open then the discussion? Or not?
Do you think so? I don't think so, because we only have half an hour.
We only have half an hour. Yeah. Oh, I thought until 7.
Adam and Kristen, come here.
Yep. Thank you.